Before we start our experiment, make sure to always be safe. That means goggles, gloves, aprons, and closed-toed tennis shoes. The materials we'll need for our experiment are 420 milliliters of water, 30 drops of iodine, 2 tablespoons of starch, 2 beakers, a graduated cylinder and a stirring stick, as well as 2 binder clips and a teaspoon. Our hypothesis is, if the size of the molecules in a solution are smaller, like in iodine, then the solution will be able to freely pass through the semi-permeable cell membrane compared to bigger molecules in the solution, like in starch. The dialysis tubing represents the cell membrane in this experiment. Now we can finally begin the experiment. Our first step was to soak 2 strips of dialysis tubing in water for an hour or more. We have already done this. Now, our second step is to make our iodine solution. You do this by pouring 210 milliliters of water into a beaker and adding 30 drops of iodine into the water. Next, create the starch solution by pouring 210 milliliters of water into a separate beaker and adding 2 teaspoons of starch into the water. Make sure to stir both solutions thoroughly until they are mixed. Now, open the dialysis tubing and using a graduated cylinder, pour 10 milliliters of the iodine solution from your beaker into the dialysis tubing. Then create a bag around the solution by tying both ends of the tube. Make sure there are no leaks. Do these same steps with the same measurements, but with a different bag in the starch solution. Once you have completed that step, use the binder clip or clamp and attach the bag of the iodine solution into the beaker with the starch solution, and the bag of the starch solution into the beaker with the iodine solution. Make sure they are fully immersed in their respective solutions. After 15 minutes of the solutions being submerged, record and analyze the data and observations after removing the bags. Our results were, when the bag of the iodine solution was placed in the starch solution, the iodine diffused out to the starch solution, causing the contents in the beaker to turn purple. And when the bag of the starch solution was placed into the iodine solution diffused into the bag, causing the substances in the bag to turn purple. In conclusion, the dialysis tubing demonstrated the cell's membrane semi-permeability due to the fact that the iodine was able to diffuse through the bag to affect the starch solution, but the starch solution was not able to diffuse through the bag and affect the iodine solution. Our hypothesis was proven correct because the iodine has smaller, less complex molecules than starch and is able to freely pass through the dialysis tubing in the cell membrane in this experiment, while the starch's bigger, more complex molecules could not cross. This is proven with the fact that while the starch solution inside or outside the bag turned purple in both beakers, the iodine solution remained a constant color, showing that the starch was not able to diffuse through the bag and react with the iodine solution. Make sure to clean up your station after the experiment. Thanks for watching.